Alrighty. So for this poster, my name is Tram, and I will be presenting this poster over my research of Dr. Baer's research over molecular phylogenetics of Central American shrews. So as we all know, DNA is an important discovery for scientists to understand anatomy, function, and how it plays its role in evolution. So the advancement of biotechnology, it enables researchers to basically explore and discover new species from DNA extraction. So at the moment, I'm currently assisting Dr. Baer to understand shrew in the region of Central America and how there's more diversity of this species than it has been described. So the research comprised of three main techniques such as PCR, gel electrophoresis, and DNA sequences to analyze sequences of DNA that were extracted from shrew. So as you can see in this figure, this is an example of the Blarina brevicauda, which is commonly known as the northern short-tailed shrew that can be found in Central America. And for this slide, this figure represents a phylogeny that is based on the 16S gene of the SORX sequence. So at the top is the Blarina brevicauda, which was taken from GenBank. And GenBank is basically a database where scientists can freely upload DNA sequences and they are made publicly. So our data, it consists of cryptodes, and at the very bottom of this phylogenetic tree, it shows our SORX sequence. So because SORX sequences are very unique, they are most closely related to SORX sosari, but we can't really classify them as being of this species specifically. Since the branches are also scaled to divergence and not time, this tree shows multiple species of cryptodes and SORX. So divergence simply means that evolution, it occurs when two groups of the same species evolve within their groups to adapt to different environmental stresses and social pressures. So the occurrence of multiple species means that there are a lot of divergence with different species of shrews. So I do believe that further research is still required. So we need more species of cryptodes to see if there are any relations or new species of shrew from the sequences. And from what we know so far, based on the progression of this research, it is unclear at the moment whether our SORX sequences represents unidentified species. So, and I just want to thank Dr. Baer for providing guidance for making this research project possible. And if you have any questions, you're welcome to ask them now.
Yeah, like, is there at least a current species count of these shrew? Like, do they know at least right now approximately how many there might be?
So yeah, there's some. So as I mentioned before, from the example above, that's one species in Central America, but they're like, I think there's still a bunch of other species because they're all different, have DNA sequences. So I think like as mentioned before, the SORX sequences are mostly closely related to the SORX sosari, but we can't really say that the SORX sequences are that species. So that's why we still need more data to be able to identify if they are the species or if they're a new species.
Okay.
Yeah.
That's pretty cool.
Thanks.
Yeah,
Thank you for the question.
Yeah.